To analyze next-gen sequencing data for clinical research using LaserGene, start by setting up an assembly in SeqMan Engine. Whether you're working with whole genome, transcriptome, exome, or other resequencing data, just choose your project type, name your project, then load your reference sequence or genome template package, followed by your read data. LaserGene supports assembly and analysis of data from Illumina, Roche 454, Ion Torrent, and Pacific Biosciences. You can also specify sample names for multiplexed or multi-sample experiments. Review your assembly options, and then click Assemble. After assembly, you can bring the project into SeqMan Pro to view coverage along the genome, as well as read alignment for each sample. You can also use the integrated SNP report to view a list of putative SNPs in the assembly and filter SNPs based on criteria such as depth of coverage and impact on coding features. To analyze large sets of SNPs across samples, bring your assembly into ArrayStar. Here, you can organize your assemblies or samples into different groups. In this case, I've created a control and a disease group. You can then view a list of SNPs in the assembly or a table of genes, each of which are assigned a disruption value based on the impact of SNPs within the gene. You can also filter genes and SNPs based on a variety of criteria, including the impact of a SNP on a gene and whether the SNP is present or absent from dbSNP or another SNP database. You can also filter based on which samples contain such SNPs or search for SNPs of statistical significance. For example, you can use the Fisher's exact test to look for genes with a low p-value when comparing SNPs in the diseased and control group, or when comparing SNPs between samples based on imported phenotypic attributes. You can then use your filtering results to create and compare gene or SNP sets, and then view gene ontology information for the genes of interest.